Awesome. Thank you so much for the introduction, and thank you so much for the to the organizers for setting this up. So today I'm going to be talking to you about sort of my journey in developing a SARS-CoV-2 vaccine. I'll be talking to you about a lot of the preclinical studies, as well as some of the phase one clinical data. And shortly after that, I'll open it up to sort of discussion, and we can feel free to answer questions or just chat about some of the data that I presented. Um, I think I think I'd first like to start off just talking a little bit about my career trajectory. Early in my undergraduate career, I learned, you know, uh, uh, the aspects of doing research at South Virginia University. I graduated in 2014, and I did a short term at Rhode Island College where I was teaching some high school students, in particular in two academic courses in advanced biology and also a state testing mathematics course. And I learned also a lot of, essentially, I developed a passion for teaching, which is something that sort of drives me now at Beth Deaconess Medical Center, where I'm one of the leaders in the virology group. And I'm very much involved with a group of technicians. I lead them on a day-to-day basis, and I ensure that all the work is being done, as well as I have several projects currently ongoing, and also ones in the past that I've led that have ultimately led to essentially has set up the platform to develop the SARS-CoV-2 vaccine. I've highlighted a few things in this slide. One of them, a few years ago, we developed a Mosaic HIV-1 vaccine, which is currently in large phase 2B trial in Africa, and it is also in phase 3 efficacy trials in the Americas and Europe. We additionally have developed a vaccine against Zika virus. In preclinical, our preclinical data demonstrated 100% protective efficacy. And currently the phase 1 data has actually just been published, I think about a month ago. So very good results from that study, as well as the Mosaic HIV-1. So I won't be going too much into detail about the HIV vaccines, but or the Zika virus vaccines. I want to focus more on the SARS-CoV vaccine, which has been approved by the FDA and is currently being distributed worldwide. So it's been a very wonderful journey. I think I owe a lot of the work that we've done to the great team that I have in the lab. I've published so far, just being in the lab for over 5 years. I've published in every major journal, including Nature Medicine, Nature Communications, Science. So we've been very productive on that end. And we've also been featured in several different news media outlets, including the one you see in front of you, which was published on July 17th by the New York Times. So if you'd like to learn a little bit more about sort of the journey to there from the perspective of the New York Times, I encourage you to read it. But without further ado, I think we can move forward into the talk. So I want to start off mentioning a little bit of background about SARS-CoV-2, or severe acute respiratory syndrome 2. SARS-CoV-2 is a positive sense single-stranded RNA virus. It has a single linear genome, and it's about 30 kb. As you can see in the diagram below, there are several accessory proteins, as well as structural proteins termed spike, envelope, membrane, and nucleocapsid. The main focus for the vaccine strategies is the spike protein. And one of the reasons that is so is because it is on the outer membrane of the virus particle, and it is the first site where it comes in contact with a cell. So it utilizes ACE2, that's its primary receptor. And that ACE2 is predominantly expressed in epithelial cells and in the small intestine. So you can already see where the pathology is primarily found, and why we have a lot of definition for where we see a lot of the pathology and also the outcomes for individuals who are infected. I also want to mention that evolutionarily, SARS-CoV-2 has developed this amazing strategy to produce subgenomic intermediates, and these are shown in the diagram below. Hopefully you can see my pointer. And so these subgenomic intermediates are a measure of replication. So this will become important a little bit later in the talk when I begin to talk about one of the assays that we have validated in our lab, demonstrating that we can target these intermediates to measure essentially ongoing replication. So I also want to talk a little bit about the vector-based vaccines. So these viral vector-based vaccines have been around since the early 1970s. They're designed to be replication incompetent. They're safe and immunogenic. And the technology has actually been used in several different strategies, not just in our lab, but in several different labs around the world for countermeasures and therapeutics against HIV, flu, Ebola, and now SARS-CoV-2. So one key thing of the viral vector design is that they're generated, as I mentioned, replication incompetent. And because they're replication incompetent, they will never infect your own cells. They would have to have a complementing gene that can be grown. So they can be grown in vitro, but they will never be able to replicate in your body. However, it's a great delivery method because you're able to express your antigen of interest. In particular, what we have designed is ones that have the spike protein and a modified version of the spike protein. So if we look back to July of 2020, I want to talk about the one of our first publications in Science termed SARS-CoV-2 infection protects against rechallenge in rhesus macaques. And as part of this study, we challenged a total of 9 rhesus macaques with 10 to the 6th, to the 5th, or 10 to the 4th PFUs as designated by Group 1, Group 2, and Group 3. And if you look at this, this figure here, you can see that after initial infection, we were able to measure nice high responses for ELISA binding antibody titers, as well as pseudovirus neutralizing antibody titers. So overall, these antibody titers seem to correlate really well with convalescent patients. So we were sure that these would be protective. So if we followed, the next step in this study was to look at reinfection. And the overall idea was to find out whether initial infection was able to protect from reinfection. At the time, we didn't know this. This wasn't something that was inherently obvious. And with the pandemic just starting, this was one of the questions that essentially our lab was able to answer in the macaque model. So if you look at these, the first panel in the BAL, the bronchoalveolar lavage, so that is a representative of the lower respiratory system. We can see on this top panel here, when we measure the viral loads post second infection, we can see that there is a very nice reduction in viral load in all 3 of the groups. When we compare that to a naively infected macaque, we can see viral loads peaking anywhere between 5 to 7 logs. And if you look at both the primary challenge compared to the re-challenge, you can clearly see that there's a statistic significance between the two. And we were able to show that there is a nice reduction in viral burden in the lower respiratory tract. So what about the upper respiratory tract? So the way that we measure this is by looking at nasal swabs. So here we're looking at, again, the viral loads in the upper respiratory tract, and looking at from Group 1, Group 2, and Group 3, we can actually see that there's a steady reduction in Group 1 post-reinfection, which is very significant. So you have to think about how much virus we actually gave to these in Group 1. So Group 1 received a very high dose, something that you would in nature you would not be exposed to. In groups 2 and groups 3, we're able to see a very nice reduction. When we look at every single macaque as a whole, compared from pre-challenge, primary challenge to re-challenge, we see a nice reduction again in viral load, and that is what's also statistically significant. So essentially what we showed here is that immunity acquired through initial infection protects against a future infection in the rhesus macaque model. So this was a very nice segue into how we design our vaccines. So we know that there is protective immunity. We now knew that the rhesus monkeys were able to elicit some sort of protection. And we wanted to know, well, this has to be antibody mediated. It has a potential to also be T-cell mediated. But this is why we moved into the initial testing of our prototype vaccines. And in here, we generated 6 different DNA vaccines, and these express different versions of the spike protein. So the full-length spike protein is in red, and you can see subdivided it into the 4 different domains. You have the S1, the S2, the transmembrane domain, TM, and the C-terminal domain. So we generated different vectors, different constructs with varying lengths of this spike region, including a full native spike, a SDCT, which is a delta C-terminal deleted, delta terminal transmembrane deleted, just the S1, the RBD, which is the receptor binding domain, and also a construct which will be really important down the line, which is a full-length spike with the transmembrane deleted with this 2 proline stabilizing mutations. And I'll go into a little bit more detail a little bit in a few slides about it. So we looked at the Western blots, as you see on the bottom panel, and we noticed that all these, we observed that all these vaccine constructs express, which is what we are expecting. So we moved that into rhesus monkeys. So what we did is we vaccinated several rhesus macaques with 5 milligrams intramuscular. And we also included 10 macaques for the as the sham controls. So if you look at the first panel here at the top, in the bronchoalveolar lavage, as well as the nasal swabs, we clearly see the normal viral kinetics as measured by qPCR. So we see a nice uptick of viral load peaking by day 2 and sort of self-resolving by between day 10 to day 14. In these nasal swabs, the upper respiratory, we see the same kinetics. We see a nice uptick of the infection, and then we see a nice clearance between day 10 to day 14. So what was interesting in this is if you look at the vaccinated macaques here, if you look at the ones that received essentially any of our vaccine constructs, they all had very much lower levels of viral load compared to the sham group. So all the groups or all the vaccine constructs had a nice reduction, meaning that they were protected. So this was very, very amazing to find a reduction that would essentially mimic less severe disease. And this was consistent in both the BALs and the nasal swabs. The one construct I think that was probably one of the least ones is the full length with the transmembrane deleted domain. However, even that construct, just to demonstrate the validity of these different vaccine candidates as a potential for them to be used was great to increase the knowledge. So if you look here, here's the summary of all these viral loads. So we can see reduction, and the most significant reduction, interestingly, was in the full length spike, which will be, again, important when we talk in a little bit about the vectors that we developed and that we tested in the At26 platform. Okay. So in July, I actually published one of my papers titled Single Shot at 26, Vaccine Protects Against SARS-CoV in Rhesus Macaques. This was a proof of concept study, which essentially led to the best vaccine candidate that we moved into clinical trial. So we generated 7 vaccines in collaboration with Johnson & Johnson. These include a TPA. Again, you can see here on the first panel. So you have the full length spike as we saw before. So we generated constructs with TPA, which is the tissue plasminogen activator, which has been shown in the literature to enhance gene expression. We also included the native full-length spike as a control. And then we included a few different variations of those, including a proline stabilizing mutation, as well as a cleavage site deletion. And one of the reasons we did the cleavage site deletion is because if you think about essentially what the cleavage site does is it cleaves the S1 and the S2 domains. So when you actually become infected, in a normal viral particle has that native furin cleavage site. So therefore, we wanted to mimic what the virus particle looks like, or how the immune system looks at the virus particle. So we mutated that to keep both the S1 and the S2 domains, and also in order to enhance immunogenicity. The proline stabilizing mutations are actually very important. There's a few papers out there that sort of describe why the 2 proline stabilizing mutations are important. And just to briefly describe that, essentially the spike is a trimer. So the spike actually exists in 2 conformations. So you can have either what they call a down conformation and also an up conformation. So when you have the proline stabilizing mutations, it keeps the trimer in an up conformation. Therefore, it allows more epitope domains to be exposed and also enhances immunogenicity and the amount of immune response that you can get in that trimer. So with these constructs, as I mentioned, we made them in the recombinant adenovirus serotype 26 as the delivery platform. We again tested their expression by Western blot, as you can see in the bottom panel, and all the constructs were expressing. We then immunized rhesus macaques, and then we challenged them with SARS-CoV-2 to look at the viral kinetics. So, before I show you the viral load data, you can see here, this is the ELISA binding antibody titers, as well as the pseudovirus neutralizing antibody titers. And all of our constructs by week 2 elicited high levels of binding antibody titers, as well as neutralizing antibody titers. And by week 4, these actually peaked. So one of the, a few of the constructs seem to be working really well. One of the main ones in these experiments was the full-length S.pp, which will sound a little bit familiar because, just to give it away, essentially that was the best candidate. So we already had an idea even after the first or after the initial experiments. So we also evaluated T-cell responses by ELISpot. So as you can see here, all the constructs were able to elicit high levels of T-cell responses, in particularly as compared to sham, and all these responses peaked at week 4. We also looked at ICS as measured by interferon gamma responses, and again, we saw that and what we saw was that there was not really a significant difference between the interferon gamma producing CD8 T cells. So what that does mean is that you're not going to get an amazing T cell response from any of these vaccine candidates, at least compared to sham. However, what we think is that most of the protection was going to be mediated by antibodies and not necessarily T cell responses, although T cell responses do seem to help in clearance of initial infection. So here's the viral load data for all the different constructs. So on the left panel, we have the sham control groups, and we have in the lower respiratory system, in the bronchoalveolar lavage, BAL fluid. And what you can see strikingly here is that compared to sham, one of the as I mentioned, the leading candidates, essentially the STAT-PP completely cleared infection. So all the macaques cleared the infection. Um, and in all the other groups, again, there was a very striking reduction in viral load and in essentially in severity of disease. And the nasal swabs, as I mentioned, is a higher bar to overcome. And even in that, we only had one single breakthrough in one of our top candidates, and all the other constructs had a very nice reduced infection. So what this essentially shows is that full protection in the lower respiratory tract, and we can also see near full protection in the upper respiratory tract by one of our leading candidates. Shortly after that, so after that, we actually published a lot of different papers. We've gone and covered all different topics, including severe disease in the hamster model. We've published just recently a subgenomic assay that I won't be going into detail for now. We've shown correlates of protection. Also, we've collaborated with other individuals, demonstrating how neutralizing antibodies can also be used as therapeutics and as preventative interventions. So it's been very very productive and very fruitful in the lab in terms of collaborations, as well as all the work that we've done. Um, so I want to shift gears a little bit and steer away from the preclinical trial and actually talk a little bit about the results of the phase 1/2 trial. Um, and then we can open it up to talk about a little bit about the phase 3 trial, which the results have not been published yet. However, all the data is publicly available by the FDA and in cdc.gov. So in the phase 1 clinical trial, phase 1 and 2 clinical trial, we evaluated the At26 CoV2.S, and this was the best candidate, as I mentioned, the one that has the furin cleavage site, as well as the two proline stabilizing mutations, full-length spike. As part of this clinical trial, we evaluated two doses, a low dose at 5 times 10 to the 10 viral particles, and a high dose at 1 times 10 to the 11 viral particles. So one of the most frequently, or some of the most frequently solicited events were fatigue, headache, myalgia, injection sites. One of the the worst systemic adverse event was fever. So we can see just from these descriptions that the vaccine was very safe. It had very low reactogenicity, and individuals who received it had very mild symptoms. So we had two cohorts, and in the left panel, you can see the distribution of the two cohorts. We had a cohort of 18 to 55-year-olds, and then a cohort of individuals over 65 years old, which was essentially the most critical group, as these are the individuals that are most likely to become infected and have severe disease. So as part of this study, we measured one of the first things that we measured is the spike ELISA titers, and I'll go over. This is a very busy sort of slide, so I'll break it down a little bit. So if you look at the first arm in the cohort 1A, which is the 18 to 55-year-old participants, we had essentially a control group which received 2 doses of the placebo. We had a low dose, one single low dose, and then the next dose was a placebo. We had 2 low doses, one high dose, and one placebo. and then 2 high doses. And we evaluated in looking at the binding antibody titers, we can see that there's a very nice generation of these antibody titers across all different groups. And in particularly, what's nice about this study is that we can show that even at a low dose, which is honestly better for manufacturing, for distribution, and for essentially any systemic events where you don't want to give someone a really high dose. You want to make sure that the individual is able to tolerate a variety level of doses, but particularly if they're better suited for a low dose, then in a clinical trial, we'll always divert back to a lower dose rather than a high dose, which would cause a lot more systemic adverse events. So we can see here that even individuals who receive just a single low dose compared to 2 low doses, there was almost no significant difference between the binding ELISA antibody titers. And when we looked at the individuals that had either received one single high dose or 2 high doses, again, there wasn't essentially much difference. And this is compared to a group here, which is the human convalescent serum. So as you can see, all these binding ELISA titers were either at or above human convalescent serum, which is a really nice bar to set because now we know that from initial preclinical studies, we know that these convalescent humans are able to prevent reinfection, right? So these individuals will not be able to get reinfected. So if we can mimic that with a vaccine, then that means these individuals should be protected once we scale up to larger clinical trials. And in the cohort 3, which is the individuals 65 years or older. Again, what we evaluated here, we looked at a placebo arm. We had 1 single low dose and 1 high dose arm. And in individuals that are 65 years and older compared to convalescent plasma, we were able to see we either at the same levels of binding ELISA titers or at slightly levels. So protection in older adults, of course, the amount of immune response, I should say, in older adults is a little bit lower compared to younger individuals. However, we did see that we were able to elicit nice high binding antibody titers. In the same story in the lower panel here, where we looked at the neutralizing antibody titers, we were able to see a similar story as the binding antibody titers, where we were able to elicit high levels of these neutralizing antibodies in all individuals that were either at or above compared to human convalescent serum. And it was a similar story as we had seen with the binding antibody titers for the individuals 65 years or older. So next, we also, as part of this clinical trial, we also looked at CD4, CD4 T cells, as well as CD8 positive T cells. So what some of the major points just from this panel right here is that 83% of the participants had detectable T cell responses by day 15. So this is in the similar fashion as the previous one, where we had the placebo arm, the low dose, and then a single high dose arm. And we looked at both cohorts for the 18 to 55-year-olds, as well as the cohort of the older than 65-year-olds. And we were able to see that 67% in cohort 3, which is the 65-year or older, elicited T cell responses. And they were skewed towards Th1 T cell responses rather than Th2. So overall, the CD8 T cell responses were slightly lower, but again, this sort of goes into the correlation that T cells seem to play a role in this protection. But again, more studies need to be done to sort of evaluate how much these T cells are driving that initial clearance, or how much they're helping in protection overall. And I think I just want to sort of conclude with that phase 1 trial. I want to open it up to hopefully everyone who has questions. This picture here is actually of our lab a few years back. As you can see, Larry's actually right over here. So I definitely want to thank all the lab members, my PI who's in the center. All the work that we've done is because of his leadership, and again, my team who's been really instrumental in developing and generating a lot of this data. And with that, I'd like to take any questions. I don't want anybody to be shy.
But when you started the clinical trials with humans, were there any challenges that you guys experienced, or if there were any, and if you had to make any adjustments?
Yeah, that's actually a good point. So I didn't touch much about the transition. So preclinical studies, a little bit more simple. So we have a lot more control as to what we can do. And again, these are challenge studies. So, we're challenging the rhesus monkeys that we use, which is something we cannot do in human clinical trials. So initially, a lot of the recruitment efforts were done by one of the groups in our lab. They're the clinical trials unit, and it wasn't difficult to actually get participants, because individuals were really interested in signing up for the clinical trial. They wanted to be vaccinated. They wanted to contribute to essentially ending the pandemic, and they very much do to this day when we have long-term follow-ups. So yes, there was a lot of difficulty, primarily because a lot of the individual buildings, a lot of the research labs had actually shut down. A big portion of the hospital had actually slowed down. And the reason being is that they were expanding, unfortunately, they were expanding ICU units. They were expanding places where they couldn't fit individuals with severe disease, individuals who, a lot of them unfortunately did not make it. So yeah, there was a lot of fear. There was hesitation as well. But again, because of the brave participants who wanted to contribute, we were able to move the study forward.
I have a question about how did you handle, since you took a lot of participants who are really old, how did you handle their other medication needs? So they would probably be taking either diabetics or any other conditional medicine. So did that kind of cause you to have some issues when you were giving them the test vaccine, or was it okay?
Great questions. So the candidates are actually screened. So that's why we have the entire CTU, the clinical trials unit. So they are screened essentially, and the questionnaire is very long. Are you currently taking any medications? Do you currently have any sort of um, uh, 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 physiological disabilities? Do you have cancer? Do you have any diseases? Things like that. And we had to actually screen out a lot of individuals, because our trial didn't necessarily, we didn't necessarily look at individuals who had other other diseases, or other problems. And we also didn't want to skew our data. So we wanted to make sure that the individuals that we recruited were, hopefully as fit as can be, off the top of my head, in the phase 1 and clinical, in the phase 1 trial, there weren't really many individuals who had to take some, extraneous amount of medication, just your regular medications. And in this, that's why we listed sort of all the individuals and what exactly they were taking at the time. This became a little bit more complicated in the phase 3 efficacy trial because we wanted to have a more robust approach. We wanted to make sure that we included all individuals. And regardless of whether they were on any other medication, their responses were the same. Of course, we cannot take certain individuals, for example, those that are compromised. So these individuals will not be able to qualify for our trial just because of, again, these individuals have prove a whole different subset, and they answer a different question, but they also provide a little bit more challenge than we sort of wanted to. And, out of the entire cohort that we evaluated, the phase 1 was only a small subset. Phase 2, we increased to the 1,000-fold. And in the phase 3, which was a multi-city, like worldwide, essentially, it was not just our unit, it was not just our site participating, but several sites around the world. That there were over 40,000 participants. So, all of them had a lot of different, they come from all different walks of life. And what you saw summarized, or what you can actually look up summarized in the FDA approved data, is essentially you can see that everything that we looked at, and they all seem to respond equally to the vaccine.
Thank you.
You touched on this briefly about running the phase 3 trial regarding the efficacy. And my question kind of revolves around some of the misunderstandings some people have regarding the actual numerical value. Because when you take a look at the mRNA vaccines, they report a like 95% efficacy rate, whereas Janssen provides a 76%. Some people assume that one's better than the other. So can you touch on that sort of disconnect where people have between the efficacy rates?
Definitely. So what I can say is be careful what you listen to in the media. One of the reasons being is that not all the trials were done in the same fashion. Not all the trials were done in the same sites. The individuals are also different. The participants are different. So you have to take a look at the time frame of when the trials were conducted. So what I want to say is that if you look at the Pfizer Moderna, they were done very early on in the pandemic. The common circulating strain was the original Wuhan isolate. So the vaccine that we designed, as well as the vaccine that all the other labs have designed, was based on the original strain. So now that we have smaller outbreaks of these variants, the question sort of comes, well, do these vaccines protect against these variants? And fortunately, our trial was actually extending through the time that the UK and the South Africa variant was around. So we didn't sequence every single individual for that became infected, but we sequenced the majority of them, and we were able to get data based on the efficacy, the safety profile, and the protective efficacy of our vaccine against the UK variant. And that's where you see all these different numbers. Whereas in the Pfizer and Moderna, you can see that they have one single efficacy readout, which was, again, based on the time frame of when and what virus isolate was circulating. And again, I won't harbor on the numbers, again, because the clear message is, of course, that all these vaccines are safe. All these vaccines elicit robust immune response. So you are going to be protected. And regardless of which one you get, you have a very low likelihood of being in a ventilator, of having severe disease. So that's, I think, the clear point. And of course, the media sort of, sort of takes an uptake on this, and sometimes likes to take sides a little bit. But yeah, definitely, that's why most health professionals, doctors, Dr. Fauci, anyone you sort of speak to, will encourage you to take anyone that's sort of readily available. Because again, some protection that is anything above 50% is good protection. So that means you're staying safe, and that means you're keeping the individuals around you safe.
I think there's another question for you in the chat, Noe, and I could read that.
Yeah.
Did your research group find yourselves under any political pressure to advance this research, considering the social climate revolving around the virus?
Wow. These are great questions, guys. In terms of the no, one of the reasons being is that our work was not politicized, in the sense that when we first when we first started working on the vaccine, no one knew that we were working on the vaccine. Essentially, it wasn't until we built the collaboration with J&J that we actually became publicly known, and even a little bit before that, only locally. So in terms of any political pressure, I would definitely say no. We never had someone, or someone from the government or from the outside sort of reach out and sort of ask us, hey, what are you doing? How quickly can you get this done? What we wanted to focus on is, again, the data. What we wanted to focus on is advancing the vaccine as quickly as possible, however, without cutting any corners. We wanted to make sure that once we presented all the data, and ultimately what we did to the FDA, to an external panel of scientists, not ones that are part of the government or any political group, that they would be able to look at our data and make their decision based on that. So you can see we had a very straight transition from initial inception of the idea for the vaccine all the way through in our phase trials. We were very transparent about it as well. So all the data was actually publicly available before it was formally published. And we continue that trend even today. You can see a lot of the collaborative papers that we've, or the manuscripts that we've written up. They can actually be found on bioRxiv. They're publicly accessible, and they have not been formally accepted by any journal, so they don't own any of the rights to it. And again, I think, I guess, in terms of the politics, right? You always think that politics drives a lot of what we do, but what we wanted to do was harper on and make sure that we never lost sight of the science.
No, I just want to jump in real quick. So number one, great presentation, and I'm really proud of you and all your contributions. It's really nice to see your career take off. I have a lot of questions, but I'll just narrow it down to two. So since it seems like you kind of hinted that T cell responses may be involved in some way in addition to antibody-driven responses, did you guys do any T cell depletion studies to rule that out, and particularly in an animal model system? And then the second related question is, since this is an antibody-driven process, and we do have these new circulating variants that are floating around all over the place, some of them are more virulent than others, do you know exactly where those neutralizing antibodies are binding as part of that spike protein, right? So, and then in addition to that, obviously the follow-up question is, are any of those circulating variants, those mutations, occurring right around where those neutralization epitopes are?
Good. So to address the first question, so in terms of the T cell responses, we know that there is some effect, and we actually did look at the correlates of protection. We actually published that in macaques, and there is, we did see that there was T cell mediated helper cells, primarily CD4 T cells, that were aiding in the clearance of initial infection. So we did publish that paper in the correlates. I didn't go into details about it, but most of the protection really is antibody mediated. So I think a lot of that still has to be teased apart. We only did a small cohort of animals as a subset of those correlates. We didn't do it a massive, and then we also don't know if that is recapitulated, but we assume it is in humans. So again, I think more information regarding what the role of T cell plays, not just CD4s, but also CD8s. Right, so we need to do, and that's sort of one of the key things. We need to do studies on both depletions of both. We need to be careful how we deplete those, because some of these depletion drugs actually will target a lot of other innate immune stimulators or other innate immune cells, I should say. And so we, again, that's something that needs to be sort of more dealt on. But yeah, if you have a moment, definitely read the paper on correlates of protection of SARS-CoV-2, and you can see the small cohort, and you'll see how the protection essentially, there is some indication that there is some T cell mediated, and that's one of the nice robust things that we like to say about vector-based technologies, where they elicit nice T cell based responses, which have the potential to enhance the overall immunogenicity, right? And so hopefully that answers your question. Yep. All right. And also for question 2, so for, sorry, can you say that, repeat the question again?
Yeah, so the question was based on, and the reason like I brought it up, so Billy had asked a question earlier about, you know, when you're comparing these different vaccine trials, right, with Moderna, Pfizer, Johnson & Johnson, it's not really comparing apples to apples, because the Johnson & Johnson was undergoing when the Brazilian, the African variants, they were prevalent and all over the place, right? So, so because of that, and because those numbers are down a little bit lower than some of the other ones, I'm just curious if some of the mutations that have occurred within the spike gene, if those are around the neutralization epitopes that are being generated by these vaccines.
Yeah. Oh, yeah. That's actually a good question, and that actually was one of the reasons why I think the biotech company was, I think their name was Eli Lilly, actually stopped their antibody production, because what they noticed is that based on the variants, that essentially their antibodies were no longer neutralizing, and they were not protective against the new variants. So there are some nice literature out there about the different epitopes where SARS-CoV will bind. Again, it's still random. There needs to be a lot more information. So I don't know exactly where our specific ones, we haven't done epitope mapping to look at where our specific vaccines, where our specific vaccine elicits the most, and the types of neutralizing antibodies, as well as general breadth of antibodies. So a lot of these initial neutralizing antibodies that were developed have been shown to actually functionally not work as well anymore because of these variants. Now, if you look at the genetic sequence of the spike, so they primarily bind on the spike, but there are of course other sites where you can elicit neutralizing antibodies. But just speaking about the spike, these mutations are widely different. They're either multi amino acid mutations that essentially give you a whole new, almost like a whole new spike, that the initial, that when you design the initial antibodies based on the initial infection from the original circulating strain, they no longer function as well. So for therapeutics, definitely this was a great strategy. Convalescent plasma, great strategy. But I think moving forward, the antibodies you're going to be hearing a lot less about, and also ones that are or have been in clinical trials are probably not going to move forward. As of right now, because of the state of the world, a preventative vaccine, one that generates multifaceted approach, different types of antibodies, and it can be, both neutralizing and binding, are better than sort of designating a cocktail or making one or two neutralizing antibodies that will completely neutralize. I don't think that will be feasible. And this is obviously knowledge also that we have based on the flu, right? So a lot of when we receive the seasonal flu vaccine, it is typically an inactivated version or a attenuated version of the current circulating strains. So you don't really hear too much about antibodies because of the rate of mutation on the influenza virus.
Any more questions for Noe?
Yeah, I kind of had another question. So obviously the J&J is an adenoviral vector vaccine, but when compared to the AstraZeneca, since that is also adenovirally a vector vaccine, was there a necessary reason why J&J decided to give this vaccine as a single dose as opposed to AstraZeneca, who is approved in other countries as a 2-dose series?
Good question. Yeah, so one of the reasons is actually, so we didn't want to put all our eggs in one basket. So we actually had a phase 2 trial that looked at both a single dose as well as a 2-dose, and including some of the early phase 1 clinical trial where I showed you that there was a 1-dose versus a 2-dose. For us, there was no significant difference between either giving 2 high doses, 2 low doses, or 1 single dose in terms of immunogenicity. I'm not sure why. I haven't looked too much at it's a chimp ad, by the way, the AstraZeneca Oxford vaccine. It's a chimp adenovirus, very similar. Ours is a rhesus adenovirus, but again, the serotype makes a big difference, whether it's human makes a bit different. So ours is a human adenovirus serotype 26. So all these adenoviruses functionally are slightly different. They're able to elicit different responses. So from our early data, we actually found that a single dose was essentially enough to essentially mediate the protection in the preclinical trials, and in the phase 1 elicit a nice breadth of binding and neutralizing antibody titers. And so we decided to go with the 1 single dose and move forward to FDA approval because the 1-dose strategy was essentially going to be more feasible for distribution. It was going to be easier to access. It was actually going to help individuals who cannot meet or who cannot get 2 vaccines, who cannot, go from one site to another site, or who cannot drive, back and forth, individuals who are older, individuals who live by themselves. Very much our approach was to design a vaccine as a single dose for the world, not just here in the US where it might be more accessible, where it definitely is more accessible, but in places like Africa, in places like Asia, in areas where you don't have the infrastructure, where you don't have all the issues that we've been hearing in the media about ultra cold temperature, all the news about the distribution, and even for just the individual who doesn't like getting shots, 1 single dose, 1 single shot, and you're done. That was definitely one of the ideas that we had moving forward. So we did look at the 2-dose approach, and since there was really no difference between the two, we decided to stick to the 1 dose. Although I will say that if we move forward with the 2-dose, maybe we would have had a better efficacy phase 3 readout that would drive our efficacy to what, 85, 90%. I mean, that's obvious. When you give a single dose as a prime and when you give a second dose as a boost, that boosting response is able to essentially give you that better protection. But that's not to say that that won't happen in the future. But I think for the current time being, we definitely wanted to focus really heavily on that worldwide single dose vaccine that will hopefully, which is hopefully now and will, keep in the future, help out and help us get and move forward from this pandemic.
Thank you so much.
Any questions in the chat?
All right, if